Hey everybody, we are on to the final topic of the semester, and that topic is muscle tissue. So let's get started by identifying the three basic types of muscle tissue. There are only three, a mere three, and we see all three of them here on this screen. We've got smooth muscle tissue, which is this type at the top picture here, usually found in hollow organs like blood vessels, digestive tract organs, urinary reproductive tract organs, airways, other places too. We'll talk a little more about it later. Down here we got skeletal muscle, which, as its name implies, is attached to and pulls on bones. And then over here, bottom right hand side, we got cardiac muscle, which is found solely in the wall of the heart, contracts to generate the force to move blood through your circulatory system. So those are our three basic types of muscle tissue. Let's rock through them really quickly, starting with skeletal muscle. Skeletal muscle, like its name implies, is going to generally attach to bone. Now, not all skeletal muscle attaches to bone. Think about the muscles that move your eyeballs, muscles that let you smile, those sorts of things. Skeletal muscle, though, is generally what we think of when we talk about muscle. If you ask a non-anatomy student about muscle, they will think about skeletal muscle before they think about cardiac muscle, before they think about smooth muscle. So, skeletal muscle, the visible muscles we see here on Mr. Schwarzenegger, pulling on the bones in your body. Now, skeletal muscle is mostly voluntary. Some exceptions: reflexes, the action of your diaphragm, the contraction of the anal and urethral sphincters. Those can all be exceptions. Now, muscle tissue cells are often called fibers because they are long and skinny. We know from lab they have a lot of these guys. They have a lot of nuclei. They are also not unlike this creature in that they are striped. And the stripes that we see are referred to as striations. And the striations result from the orderly repeating arrangement of the two proteins involved in contraction, which are known as actin and myosin. So more on them later. Okay, that is our overview of skeletal muscle. Let's do a quick overview of cardiac muscle, which is found in this heart, human heart here, in the wall of the heart. The heart is mostly cardiac muscle. It's got other tissues too, but it's mostly cardiac muscle. Now, cardiac muscle tissue is unlike skeletal in that it is involuntary. It is also different from skeletal muscle in that the cells are not long and skinny, they are short and wide, so we don't always call them fibers. Sometimes people do. It's okay. Now, there are striations here. They're a little bit harder to see, but you can make them out right where I have the cursor here going horizontally across the screen. And these striations, of course, are the result of the orderly repeating arrangement of the contraction proteins actin and myosin. Now, also in cardiac muscle, we see these little reddish structures here running perpendicular to the cells, parallel to the striations. These guys are called intercalated discs. They consist of two things. They are going to consist of gap junctions, which are going to let the heart work as one big unit by electrically coupling all the cells. And the intercalated discs also are going to contain desmosomes, which are going to strongly physically anchor the cells to one another. All right, that is the second type of muscle tissue that we're doing, and we are doing three. lastest but not leastest is smooth muscle tissue. Like I said earlier, found in hollow organs like blood vessels, found in digestive organs like the esophagus, the stomach, the intestines, the ureters, the urinary bladder, as well as places like the trachea, the bronchi, the vasa deferentia and the male repro system, the uterus and the female repro system. Smooth muscle is rather ubiquitous around the body. Now the fibers are involuntary. They're called fibers. They're involuntary as well. And they are non-striated. In fact, that's where the word smooth derives from. They're not striped. They do have the contraction proteins, actin and myosin, but they don't have the same orderly arrangement that gives us those stripes that we saw in the other two tissue types. Okay, so we got our overview of the three different varieties. Some basic things about muscle tissue. Muscle tissue is excitable. What does that mean? It means that they can conduct action potentials. When neurotransmitters bind to muscle cells, they can conduct action potentials. That's what excitable means. Now, muscle cells are also contractile. They have contractility, which means they can shorten and do so in a powerful fashion. They're also stretchy. In other words, they're extensible. Fancier word for stretchy there. And when we stretch them and we let go, they're elastic. So they're like rubber bands as opposed to chewing gum. When we let go of them after they've stretched, they come back to their original shape. So these are 4 characteristics of muscle cells, a C and 3 E's, that we should be aware of. Now, what we are going to do from here on out, we are going to talk about skeletal muscle primarily, okay? You'll get more on cardiac muscle and a little bit here and there on smooth muscle when you get to A&P number 2. So general function of muscle tissue include movement. Now this is a function of all of them, right? Movement. Skeletal muscles move the body, whether it's through the air or whatever, or anywhere. They also allow you to manipulate things. Skeletal muscles are often playing a role in supporting joints. Like we see here, the muscles of the rotator cuff that you know from lab that support the joint between the humerus and the scapula. Skeletal muscles are also extremely important for posture. And when we see posture, I even mean simple things like just literally keeping your head up, like not letting your head fall down and not letting you tumble to the ground. So not just like perfect straight up, straight back posture, but just not falling down. Last but not least, muscle cells are generally pretty inefficient at turning their energy into motion, so they lose a lot of energy as heat. So heat production, and we'll give another word for that. The other word for that is thermogenesis. That is another function of muscle tissue. Think about shivering when you're really cold. That is a prime perfect example of thermogenesis. All right, some basic functions are done. Let's look at the general anatomy of a skeletal muscle. So this whole guy here is a skeletal muscle. There are muscle cells in it, there's connective tissue, there are blood vessels, and there are nerves. The entire muscle is wrapped up in this dense, irregular connective tissue sheet called the epimysium. The epimysium is actually going to be continuous with the tendon that then anchors the muscle to a bone. So the epimysium wraps up the entire skeletal muscle. Then we have bundles of muscle cells. Bundles of muscle. So this is one muscle cell right here. This is another one right here, another one right here. You get the idea here. And what we have here is a bundle of skeletal muscle cells. Such a bundle is known as a fascicle, and that's wrapped by connective tissue called the paramecium. Then each individual muscle cell itself is wrapped by connective tissue called the endomecium. Now this should remind you of the basic structure of a nerve, where we had the epineurium, the perineurium, and the endoneurium. Very similar idea here. So notice there are blood vessels in between, connective tissue, not pictured are nerves, but there are nerves in here as well. All right. If we look at another view, epimecium, thick, dense, irregular connective tissue on the outside. Paramecium, still kind of thick, but not as thick. Then the endomecium is much more areolar in its nature and around the individual muscle cells, like these guys right here. I clicked too fast there. Just excited about this topic. If you think about a delicious steak like this one, generally the more delicious, the more tender the steak is, the fewer of these connective tissue investments it'll have. All right. Couple more things, generally speaking, gross anatomy wise. When we think about a skeletal muscle like the biceps brachii here, we typically categorize its attachment points. So we see this biceps brachii has attachment points. Generally, the less mobile attachment points are known as the origin or origins. And the more mobile attachment points are known as the insertion or insertions. And we see the biceps brachii here is attached to muscle via these tendons. Tendons are generally ropey and cord-like. Sometimes we have these sheets of connective tissue. They're acting like tendons, but they're not ropey, they're not cord-like. And such a structure is called an aponeurosis. Okay, wonderful. We said earlier that skeletal muscle cells are called fibers because they are long and because they are skinny. We also said they're multinucleate. They have many, many nuclei. And this is a consequence. The length, the skinniness, the multiplicity of nuclei is a consequence of their formation when you were just an embryo. So you start out with these embryonic muscle cells called myoblasts. The myoblasts are going to fuse together to make an adult skeletal muscle fiber. And because of the fusion of hundreds of myoblasts, we end up with really long muscle fibers that have many, many nuclei. Now, this sort of situation, when many cells fuse to form one larger cell, that larger cell is called a syncytium. Kind of weird pronunciation there, S-Y-N-C-Y-T-I-U-M, syncytium. Okay, fantastic. We are now going to look at a single skeletal muscle cell or fiber, and we're going to identify many of the important structures that it contains. We already know it has multiple nuclei because it's a syncytium. It was formed by the fusion of embryonic myoblasts. Now, the plasma membrane of the skeletal muscle fiber is better known as the sarcolemma. We met the axolemma on a neuron. We met the neurolemma on a Schwann cell. So sarcolemma. Lemma means covering. Sarco means flesh or meat. Now, besides the nuclei, we have some major organelles we're going to see. We're going to see a lot of mitochondria. This should totally make sense. Mitochondria make ATP. Source of energy, right? Muscle cells, because of their contractile activity, are going to require a lot of ATP, a lot of energy, so it should make sense that there are going to be a great deal of mitochondria there. We are also going to have the contractile machinery. So do you see all these cylinders in here? Each of these cylinders is known as a myofibril. More myofibrils, more muscle strength. The myofibrils are primarily composed of two proteins that we've met already. We didn't really meet them. We learned their names. The contractile proteins, actin and myosin. Now, we're looking inside the cell, right? So we're looking inside the cytoplasm. But for a muscle cell, we don't call it the cytoplasm. We call it the sarcoplasm. And there are two other things that are not pictured, but I want to point out to you and let you know they exist. Our muscle cells will have copious amounts of glycogen. Glycogen is the polymer of glucose. So glucose being a single sugar molecule, right? A polymer is when you string them together to make chains, in this case branched chains. And these branched chains of glucose in muscle cells are known as glycogen. And why do you need them? You need the glycogen to store the glucose. You need the glucose to be utilized in order for the mitochondria to make ATP. Now, what else do you need in order to make ATP? What gas do you need? You need oxygen, right? To make a lot of ATP, you need a lot of oxygen. So our muscle cells are going to have an oxygen storage protein called myoglobin. Oxygen storage protein in muscle cells. Okay, getting started with some anatomy. There is more. If we look at our skeletal muscle fiber under the microscope, we can see the beautiful nuclei again. We can see the striations as well. So notice how there's like a darker, lighter, darker, lighter, darker, lighter. The darker parts, so let me put an X or something on a dark part right here. The darker parts are called the A band. The second letter of dark is an A. The lighter parts, which have a line down the middle, let me X out the lighter part here. The lighter part is known as the I band. Notice I is the second letter of light. Now, the I band and A band, there are going to be two proteins that are contributing to them. What do you think they are? If you said actin and myosin, you are correct. Those are the two contractile proteins that we've introduced ourselves already. And in these A bands and I bands, we are going to have the contractile unit of a muscle cell. The contractile unit of a muscle cell is called a sarcomere. A sarcomere will extend from the middle of one I band to the middle of the next. So that's how long a sarcomere is right there. It's tiny. A single muscle cell is going to have millions of sarcomeres. We're going to look at them up close in detail very soon. All right, we're going to expand upon these A and I bands and the actin and myosin. Do not worry. So here's our muscle fiber up here. Sarcolemma, nuclei, mitochondria, and what we're really interested in now are these cylinders. And this cylinder is known as a myofibril. There are many myofibrils in here. The myofibrils are the source of contraction. And we've got one myofibril down here that we've expanded. So it's like we took this guy right here, chopped him out, expanded him, put him right here. Now, what I want you to notice are the blue and red things. These skinny blue lines right here, all the skinny blue lines are made of the contractile protein actin. all the skinny red lines going horizontally here. So blue lines horizontal. We'll get to the diagonal blue lines in a moment. But blue lines going horizontally are actin. Red lines going horizontally, there we go, are myosin. So we can see our actin and myosin here. And I want you to notice how there is a part right here where there's nothing but blue, and that's nothing but actin. And then there is a part that is nothing, well, not nothing, but that has both myosin and actin. And then we got another part here that is nothing but actin, okay? So I want you to be aware of that. So check it out. The region that just has actin, that is that light region we saw in the previous slide known as the I band. So I see two I bands right here. The region that contains the myosin as well as some actin. It's like we got both, then just myosin, then both again. That is the darker one, which is the A band that we saw a moment ago. Okay, now we can also see that there is this, what's the word? Zigzag. There's the word zigzag line in the middle of the I band. That zigzag line is known as a Z disc. And it is going to anchor the actin filaments, is going to anchor them. Oh, by the way, actin filaments are sometimes called thin filaments, and myosins are sometimes called thick filaments. So a Z disc, and I have another Z disc right here, are these diagonal blue lines here, zigzag blue lines, which are going to anchor the actin proteins. Now, there is also an anchor for the myosin. We see it here. That is known as the M line. Okay, we're almost done. We have two more things to name here, okay? In our A band, there is a region of overlap at first, right? Can you see that region of overlap right here? From here to here, there's overlap. Then it takes a break, and then there's overlap again. So the A band has overlap, just myosin, overlap. Now that region of just myosin, the region right here, that is called the H zone. One more thing, folks. Almost done. Do you remember in the previous slide we said there was that contractile unit? The contractile unit, the sarcomere, runs from one Z disc to another Z disc. So the machinery of contraction works within a sarcomere. In fact, in this muscle cell up here, okay, you see all these myofibrils, and this one myofibril is going to have a lot of sarcomeres. Here's one sarcomere. It would have another one next to it, another one on the other side, and so on down the entire length. So millions of sarcomeres. Now, in this muscle cell, if one sarcomere is contracting, it means all of them are contracting. So if I know what's going on in one sarcomere, I know what's going on in the entire muscle cell. Do you remember how action potentials were described as an all or none phenomenon? Muscle contraction is all or nothing as well. A single muscle cell contracts either all the way or not at all. All right, let me write that word down, sarcomere, just in case you forgot how to spell it. You can read my handwriting. Sarcomere. S-A-R-C-O-M-E-R-E. All right, a lot of anatomy going on here, but that is really good. That's what we want. Okay, let's do some more. So I zoomed in on a sarcomere here. I got my thin actin filaments, these beautiful blues here, are the thin actin filaments. Anchored by my Z disc, I've got my thick myosin filaments. anchored by my M line. I see my I band over here where it's nothing but actin. I band over here. My A band demarcated by the ends of my myosin, my thick filament. And from Z disc to Z disc is a sarcomere. Not labeled, but I can also see the region right here where in the middle of the A band where it's nothing but the thick myosin, and it's called the H zone. You might be wondering like what do A band, I band stand for? Well, A band is anisotropic. I is isotropic. You don't have to know that. It's named after how they interact with light. The other ones is the Z, the M, the H, all are named after German words, like Zwischenscheibe. So you don't need to worry about their names. They kind of make sense, but that's okay. All right. We're getting the structure of the muscle fiber. And we got to get the structure of the muscle fiber in order to figure out how it works. Hey, by the way, do you see all these little hands on the myosin? Look at those little hands. What do you think those hands could grab? What do you think these myosin hands could grab? Well, if you guessed the actin, you're right. The myosin hands can grab the actin. And you know what the myosin hands can do when they grab the actin? These myosin hands grab that actin and they pull it in this way. These myosin hands grab this actin and pull it in this way. And what happens is our sarcomere goes from being this wide to maybe being this wide. The whole thing shortens. And that's what contraction is, but a shortening. So the arrangement, especially the overlap here, is crucial for contraction. All right, more on the contractile machinery earlier. We got some slices here. If we slice through just the I band, we see nothing but the blue, thin actins. If we slice through the H zone, we see nothing but the thicker myosins. If we slice through the outer edges of the A bands, that's where we have the overlap. And the overlap is crucial because it's where the myosin can grab onto the actin, pulling it inward, contracting the sarcomere. All right, here is a relaxed sarcomere. Look in your computer or whatever you're watching this on, and look how the distance from Z disc to Z disc. Like use your hand or your hands or something to measure it. All right. Now, what's going to happen? Like we said earlier, these myosin hands are going to grab on the neighboring actin and pull them inward. Okay. And when that happens, look what happened to our distance between Z disc. It got shorter. Let's go back up a second. Look how big the I band is here. This is the I band, okay? Before contraction. Now remember, the I band was where there was just actin. Now look at the I band, got much smaller. So because the myosin pulled the actin inward, there's less areas with just actin, so our I band got smaller. Look at this right here. This is the H zone. The region within the A band of just myosin. All right, this is relaxed. Let's look at contracted. Well, in the contracted one, the fully contracted one, there isn't even an H zone anymore. There is no region where there is just myosin. There's nothing but overlap here. The only thing that doesn't change is the A band. The A band is the same size. A band stays the same, overlap increases, everything else decreases when we're doing muscle contraction. All right, couple more things in terms of stuff inside that we got to know. Got a different drawing here which shows me a few more things, okay? I got my sarcolemma. I see a nucleus here. There will be others, of course. I see some mitochondria. We met those already. I see a myofibril here and other myofibrils as well. Inside the myofibril, I do see the thick myosin filament as well as the thin actin filament. I see a zigzag Z disc, and I see an M line in the middle of my A band. Now, I also see these blue guys here. These blue guys are collectively known as the sarcoplasmic reticulum. It is basically the endoplasmic reticulum of the muscle cell. And its number one job is to store calcium. Calcium will be super important for muscle contraction later on. And we store calcium in the SR, or the sarcoplasmic reticulum. In fact, when the sarcoplasmic reticulum releases calcium, the calcium interacts with the filaments, allowing myosin to grab onto actin and to pull and make things shorter. All right, that kind of creates the question though. What makes the sarcoplasmic reticulum release calcium? Well, it's an action potential. An action potential zooming along the muscle cell is what's going to prompt these guys to release their calcium. But there's a slight problem. If the action potential is zooming along the sarcolemma, it's next to this guy, this SR, but not next to these. Luckily, the plasma membrane has tubes that go through and branch through the muscle cell. So look at these holes here. These holes are known as T-tubules, transverse, because they go across the muscle cell. And they allow the action potential to zip down and kind of like go practically inside the muscle cell, and thereby get closer to these SRs, prompting them to release calcium, which then lets the myosin and actin interact and lets contraction occur. Wow, that's a lot. We're going to go over all that again in the second video of this muscle lecture series. We're just trying to introduce ourselves to the actors, the players in this film. Okay. Fantastic. I think we got oh no, that was it. That was our last slide for today. Yes, I'm excited about that. So muscle video number 1 is done. I'll see you next time for muscle video number 2. Keep working hard. Bye-bye.